Welcome to week 6 in biology. This is an exciting time. We're looking at cells. At this point, you should have already looked at organelles within cells and their appropriate functions. What we're going to take a glance into is how these organelles work cooperatively to manufacture important products. And in this case, we're going to focus on the manufacturing of a protein. Proteins are those wonderful machines that do all of these important jobs within a cell. So this is some really exciting stuff. So buckle your seatbelts. Let's begin. Outline of what we're going to focus on. We're going to take a look inside of the nucleus, see this manufacturing of RNA from DNA. We will then take a gander at ribosomes, which are these protein making machines. You're probably a little bit familiar with them through previous material. They're these little robots that manufacture proteins constantly. From there, we take a look into the rough endoplasmic reticulum. This organelle functions in manufacturing proteins destined for bigger and better things, maybe destined outside to leave the cell and move outside. Take a look at the Golgi apparatus. This organelle functions and has a purpose in packaging and shipping these products and sending them to their appropriate locations. These are the primary focus that we're going to have for today's little chat. So remember, these cells in this case act as protein making machines. That's what we're looking at. We can create an analogy, start thinking about a cell as a factory. This factory is manufacturing a product. That product is proteins. A major and important job of all of these cells is to manufacture proteins. At minimum, DNA and ribosomes are required to manufacture protein. So this is our starting point, thinking of the minimal things that we need to manufacture protein. DNA simply acts as a blueprint. In a factory, you need a blueprint to know what product you're going to build. In this case, we have DNA as a blueprint to build proteins. This is the information. Importantly, DNA should not leave the nucleus. So DNA will actually code for a strand of RNA called messenger RNA, or simply mRNA. That mRNA can then leave the nucleus, attached to a ribosome, that protein manufacturing machine, to manufacture the final protein product. So production begins in the nucleus. The blueprint starts to get read. DNA is that blueprint. When you think of heredity and you think of genetics passing down information, the DNA is the blueprint that gets passed on from generation to generation. We have all of this 3.2 billion letters, 3.2 billion nucleotides of information stored in that DNA on 23 sets of chromosomes. Importantly, you may have heard of the term gene, maybe not familiar with what it means quite yet, but a gene is a specific shorter sequence of that DNA which codes for one specific protein. So on those 23 sets of chromosomes, humans actually have approximately 23,000 genes. A lot of information stored in our DNA. An enzyme actually reads one gene and builds an appropriate RNA molecule. So an enzyme functions to read the DNA for one particular gene to build the RNA, or that messenger RNA, that codes for one particular protein. Once this RNA is manufactured, it now holds the information that was once stored in DNA. And now the RNA can have that information, holds that blueprint for building a specific protein. So we can take a look at this cell and we see the DNA. The term that is used to describe the process of manufacturing messenger RNA from DNA is called transcription. So we see the DNA being transcribed into RNA. Then RNA can then leave the nucleus and attach to appropriate ribosomes. And this is where the important manufacturing process can begin. Remember that nucleus is surrounded by the nuclear envelope. This separates the DNA from the rest of the cell. The DNA should not ever leave the nucleus. So when RNA is made, coding for one particular protein, this is the strand of nucleic acid that can actually leave the nucleus. So it leaves the nucleus through a nuclear pore. That arrow came in a little bit early. It is okay. It leaves through the nuclear pore and eventually gets to a ribosome. There are two types of ribosomes, two types of manufacturing machines. Remember, these ribosomes are used to build the final protein product. Some ribosomes in our two types are free ribosomes, which float within the cytoplasm, that fluid-filled interior. Others are bound or attached to the rough endoplasmic reticulum. So we have a distinction between two types of ribosomes. You should notice that when you're looking at cell structures. So free ribosomes, we can see right here, represented by my poorly drawn diagram, represented as a red dot. These ribosomes manufacture proteins that are going to stay within the cytoplasm. Their job is to work within the fluid-filled interior, maybe facilitating chemical reactions, whatever it may be, but they stay within the cytoplasm. Those that are bound to the rough endoplasmic reticulum actually manufacture the proteins destined for, I like to say, a bigger purpose, some grand job, some grand role, maybe to leave the cell, maybe to function with an organelle. So we can see these bound ribosomes that are attached to my poorly drawn, once again, rough endoplasmic reticulum. So the rough endoplasmic reticulum, remember this is now the organelle that's manufacturing proteins destined for a different location outside of that cytoplasm. They'll synthesize these proteins. There might be some finishing touches. Just like with any product a factory is manufacturing, you might have to do some final touch-ups to the product, make sure it's perfect. But right now it's really synthesized that protein. In factories, remember, they probably need to package and ship their product. Ship them out to the customer, ship them out to the stores that will sell them. A cell is no different. They need to package and ship the products as well. To package and ship, the protein buds off inside of a vesicle. So we see this protein inside a budding vesicle. This is the package. This is like the box. So you can actually see the vesicle holding the protein that is leaving the endoplasmic reticulum. It's now packaged a little bit, but it's not, remember, it still has some final touches, final touch-ups that need to be performed. So it has another place it needs to go next. This vesicle leaves the rough endoplasmic reticulum and will go to the packaging and shipping department. So in a factory, you always have this packaging shipping department, puts the labels on it, puts all of the products that are destined to the same location in one big box, and then allows it to be sent out. That's the function of the Golgi apparatus. So this vesicle that left the rough ER fuses with the Golgi apparatus. The Golgi performs those final modifications, those final touch-ups that we talked about, sorts the proteins into appropriate areas. So a protein destined for one location will get sorted with other proteins destined for that same location. And it sorts them out and combines those that are going to be going to the same place. And finally package and ships the final product in a new vesicle. So what we can see, vesicles coming in from the rough endoplasmic reticulum. We can see the Golgi apparatus, and then we can see vesicles that are leaving the Golgi apparatus. So it creates a nice flow, a movement of these proteins as they're being packaged and shipped. There are a variety of final destinations as these vesicles leave the Golgi apparatus. Some proteins get packaged and shipped into other organelles. For instance, the lysosome, something we're a little familiar with, that digestive organelle. Some proteins might be exported outside of the cell. We have cells in our pancreas that produce insulin. Insulin is a little peptide hormone, a protein hormone. That insulin needs to get excreted outside of the cell. So some proteins need to be exported outside. So you can look at those that are destined for the lysosome in a little organelle, leaving the Golgi apparatus, and you can see those that are destined for export. Important differences here. So we went through this flow of information, starting with that DNA, moving to manufacturing RNA, moving to the ribosome, rough endoplasmic reticulum, in these vesicles to the Golgi, and then packaged into vesicles that can leave the cell. So what we should be able to do at this point is describe the synthesis of a protein that will function in the cytoplasm, in other organelles. We talked about the lysosome as an example, and those proteins that are going to be destined to go outside of the cell. When you're focusing on your discussion board assignment, I want you to focus on proteins that are destined to be exported outside of the cell. The next few parts, the next few videos that you're going to watch are discussions on the endosymbiotic theory, and then finally transport of material in and out of the cell. So movement of molecules in and out of the cell. So hopefully this is some good information. Hopefully you enjoyed understanding how your cells work, because this is constantly happening over and over inside your cells. Every cell in your body is manufacturing proteins that are going to do some important job. So with that, I'll leave you and let you work on the rest of your material. Good luck.